Okay, so we're going to go ahead and get started. We're going over chapters 6 and 7 today. So they're not long chapters, but they actually have a lot of information. So in chapter 6, we're going over photosynthesis. So basically how plants make food from the sun. And then in chapter 7, we go over cellular respiration. So essentially how animals or other consumers use the energy that plants produce, or plants convert into usable energy that they can use. So in chapter 6, again, we're going over photosynthesis. And so there are two main types of organisms on the earth. So the autotrophs make their own food, and they use energy from the environment and inorganic carbon sources such as carbon dioxide. So these are what we call producers in chapter 1. So the other type of organism is the heterotrophs, which heterotrophs get energy and carbon from molecules that other organisms, such as autotrophs and also heterotrophs, eat other heterotrophs. So they're using energy and carbon from molecules that other organisms have already assembled. So all heterotrophs get their energy and carbon either directly or indirectly, though, from autotrophs. So again, so autotrophs get their energy from the environment and inorganic carbon sources, and that's where they also get their carbon. Whereas heterotrophs get their energy and carbon from molecules that either autotrophs or other heterotrophs have already assembled. So one of the main ways that most autotrophs make their own food is through photosynthesis. So in photosynthesis, plants and other autotrophs capture light energy, and then they use it to build sugars from water and carbon dioxide. So there's an increasing demand for biofuels, and biofuels are oils, gases, or alcohols made from organic matter that's not fossilized in order to reduce demand for fossil fuels. So biofuels are heavily subsidized by the government, but much of the material currently used for biofuel production consists of food crops. So things like sugar and corn. So this is typically very expensive, and this is why it's subsidized. So it also damages the environment, and because it uses generally food crops, it's going to compete with our food supply. So it drives up food prices too. So biofuels are mostly made from corn, soy, or sugarcane. So making biofuels from other types of plants that wouldn't have an impact on our food supply is one alternative. But alternatives require additional steps to break down the higher cellulose content. So the reason why generally corn, soy, and sugarcane are used to make the biofuels is they have lower cellulose content. And cellulose is just something that's harder to break down. So remember, cellulose is a structural carbohydrate made from glucose subunits. And so if you can break down cellulose easily and cheaply, then you would be able to harvest the energy and the glucose that's in the cellulose for biofuels. So research is being done to find cost-effective ways to break down the cellulose in fast-growing weeds such as switchgrass. And so these weeds are very abundant, and it wouldn't affect the food supply because we don't eat switchgrass. So the one thing though is we don't eat the grass, but what effect would this have on the food supply of grazing animals such as cows? So there's always going to be these there's no magic bullet to solving these problems. There's always unintended consequences for anything that you do. So going back to photosynthesis, visible light is what drives photosynthesis. So this begins when photons are absorbed by photosynthetic pigment molecules in the plants. So photosynthesis converts light energy to chemical energy. So plants harvest the energy and light and store it in the chemical bonds of sugars. So light energy travels in waves and is organized as photons. and photons are just energy packets. So as you can see in the black bar, so right here, sorry, so in this black bar, visible light is only a very small part of visible spectrum. So this down here is expanded, but the visible light spectrum is only this tiny little area of the light spectrum. And so the black bar is the entire set of wavelengths that are emanating from the sun. So the wavelengths of visible light fall mostly between the 400 and 700 nanometer wavelengths. So other forms of electromagnetic radiation such as X-rays, ultraviolet radiation, radio waves, and microwaves, these have shorter or longer wavelengths than the visible light spectrum. So a wavelength is just the distance between the crests of two successive waves of light. So if you look down here, light travels in waves, and if you measure the distance from one crest to another, that's the wavelength. And so as you go, at least in the visible wave or spectrum, shorter wavelengths are the purple range, longer are the red range. So we see these different, we see light as particular wavelengths in the 400 and 700 nanometer range, as these different wavelengths are different colors. So the shorter the wavelength of electromagnetic radiation, the higher the energy. So therefore, purple light has higher energy than red. So UV light has higher energy, or has high enough energy to disrupt the bonds in DNA. And so this is why you're wearing sunblock to block the UV light from hitting our skin. So the disruptions in DNA that are caused from UV light are what actually lead to skin cancer. And so you can see here, as you go from red to purple, the wavelengths get shorter, and ultraviolet radiation is even shorter wavelength than the visible spectrum. So it's very high energy. So again, when we go from right to left, from longer to shorter wavelength, you're also increasing the amount of energy that is in the light. So pigments are molecules that absorb light of particular wavelengths. So photons are not captured by a pigment. Photons which are not captured by a pigment are reflected as its characteristic color. So therefore, chlorophyll A, which is the main photosynthetic pigment, this absorbs violet and red light. And so therefore, it's going to appear green because that's the light that's reflected instead of being absorbed. So this is why most plants appear green, because the chlorophyll is reflecting the green light. So accessory pigments in plants also absorb additional wavelengths. So some of the accessory pigments whose color is familiar to us are found in high amounts in roots, fruits, and flowers. So for instance, beta-carotene is especially high in carrots, and this is what gives them their orange color. So carrots, which are orange from the beta-carotene, what light is going to be reflected? So the light that's reflected in carrots is going to be orange. So sorry, I wasn't looking at the note. Is the audio still muffled? Okay. I'm going to have to try and switch lines then. Let's go ahead and take a 5-minute break, and I'll try and use a different phone. So come back at 7:15. Okay, is the audio any better? Can people hear me? Okay, good. Okay, so let's go ahead and keep going then. So as I was saying, beta carotene reflects orange light. Some other different colors, zeaxanthin in corn gives it the yellow color. Lycopene in tomatoes is red. And anthocyanin in violets is what gives the blue color. So collectively, photosynthetic pigments are capable of absorbing almost all visible light wavelengths. So plants don't just have one pigment, they usually have multiple pigments to take advantage of all the different wavelengths. But the ratio of the different pigments is what gives the plants their specific color. So for instance, a green plant, which has chlorophyll A, which gives it the green color, that just means it has mostly chlorophyll A. It's going to have all the different pigments, or many of the different pigments, so that it can pick up different wavelengths. So it doesn't want to miss out on the green light that's being reflected by chlorophyll A. So both chlorophylls and carotenoids are present in leaves, and chlorophylls break down before carotenoids in dying autumn leaves, and this is what reveals the yellow colors. So red fall colors are caused by anthocyanin pigments, which are synthesized by some autumn leaves, and again, this produces the red color as the chlorophyll breaks down faster. So this picture shows a normal green aspen leaf on the right, and an aspen leaf during the fall after the chlorophyll is broken down. And you can see the yellow carotenoid. So absorbing a photon excites the electrons in the pigment and boosts them to a higher energy level. So once an electron is excited, it'll return back to its unexcited state quickly, and the energy is released as heat unless it's utilized. So photosynthetic cells can capture and use this energy as it's emitted from electrons when they return to a lower energy level. So when the energy is passed to a special pair of chlorophylls, the reactions of photosynthesis begin. And so this slide isn't very descriptive of what's going on. We're going to get into a lot more detail of how the electrons go to a higher energy level and then skip from the chlorophyll and move down the electron transport chain. So I'm not expecting you to understand exactly what's going on here, but you'll understand by the end. So the light trapping part of a pigment is an array of atoms in which single bonds alternate with double bonds. And you can see that here. These are two. Here's chlorophyll A, and then here's beta carotene. And you can see that you're alternating double bonds with single bonds. And that just captures the energy and transfers it to the electrons to knock them to a higher energy level. So these arrays easily absorb photons, and the pigment molecules function a bit like antennas. So you can see that it sticks up to catch those photons. So in 1882, botanist Theodor Engelmann identified the main colors of light that drive photosynthesis and he did this by looking at photosynthetic algae. So the algae is green, so from what we just went over, if it's green in color, what color is not being absorbed? So what color is being reflected in something that is green in color? That's right. And so we know that this means that the colors red and violet, which I think on the next slide I have the spectrum again. And in the spectrum, green is in the middle, and red and violet are on the outside. So red and violet are going to be the colors that are being absorbed because green is reflected. So it's the colors that are not being reflected that you should know are being absorbed. And you can see that here. So you can see that violet is on one side, red is on the other side, and green is in the middle. So if you're reflecting green, the other two ends of the spectrum is what you're absorbing. So it was known that photosynthesis caused the release of oxygen, but in 1882, scientists didn't have any way to measure this release of oxygen. So Engelmann came up with a very creative method of measuring the oxygen that's released in photosynthesis. So we laid a piece of algae across a drop of water and shined the spectrum of colors on it by splitting the light with a spectrum. So you should have learned this probably in high school. So spectrum breaks white light into its various colors or wavelengths. And so you can kind of form a rainbow from the white light using the spectrum to break the light. So he then utilized the growth of bacteria that require oxygen to determine which part of the algae released the most oxygen. So thereby he measured which wavelength caused the most photosynthesis. So it's kind of hard to understand what's going on in this picture, but if you look, this is the piece of algae. So this green is the piece of algae. And he laid it across a petri dish with water in it that had bacteria. And the bacteria needed oxygen to grow. And then he used a prism to break the light so that it formed the spectrum across this strip of algae. And then looking at this, you can see which wavelength of light actually caused the highest release of oxygen. So he utilized the growth of the bacteria that require the oxygen to determine which part of the algae released the most oxygen. And so this indirectly measured which wavelengths caused the most photosynthesis. So as you can see, the most growth is at the violet end of the spectrum. And one part of that is because, remember, the shorter the wavelength, the higher energy that wavelength is. So violet is going to be much higher energy than the red. So there's also quite a bit of growth on this end of the spectrum and very little growth in the middle. And the reason why is because chlorophyll A, the light's being reflected. So remember, it's a green piece of algae, and so therefore the green light is going to be mostly reflected. So this allows Engelmann to conclude that green algae absorbs red and violet light. So his results constitute one of the first absorption spectra. And the picture on the right shows absorption spectra of chlorophylls A and B, beta-carotene, and 2 phycobilins. And these absorption spectra, so this is the picture on the right over here, this shows the peak amount of photosynthesis for each of these colors. And so it shows the efficiency with which these pigments absorb different wavelengths of visible light. So if you look at the absorption spectra of phycoerythrobilin in the middle, so that's this red one right here. So this is the picture. Specialized to absorb light between 500 and 600 nanometers. And the reason why is because this is the only wavelength of light that penetrates water. So if you have kelp growing deep in the ocean, it's going to be specialized to absorb light that can actually penetrate deep into the water. So if anybody who's ever gone diving before, the deeper you go, the less saturation colors have. And that's because not all light penetrates deep into the water. And mostly it's the red light. So a lot of times divers will take down light to really get that color. Because otherwise everything just looks kind of bland. It's not very saturated. So this is why kelp has a brown color, because the pigments absorb the greenish colors between 500 and 600 nanometers, and it reflects the other colors. So what light does get down there, or actually if you pull kelp out of the water, all the light's getting to it and the other colors are being reflected. So the flow of energy through the biosphere starts when chlorophylls and other photosynthetic pigments absorb the energy of visible light. So in plants, some bacteria, and many protists, this energy is what ultimately drives the synthesis of glucose and other carbohydrates that are used by almost all living organisms. So again, it's this conversion of light energy into chemical energy. So it converts the light in the energy in the photon into energy that's stored in carbohydrates. So photosynthesis occurs in chloroplasts, and plant chloroplasts have two outer membranes and are filled with a semi-fluid matrix called stroma. So stroma contains an inner folded thylakoid membrane, which forms stacks of discs, which are thylakoids connected by channels. So chloroplast is an organelle specialized for photosynthesis, and chloroplasts are found in plants and some protists. So the overall formula for photosynthesis, and it looks very complicated, but it's actually really not, is carbon dioxide plus water and light energy forms glucose and oxygen. So you don't need to remember the numbers here, so the 6 carbon, just remember the word. So carbon dioxide plus water plus light goes to glucose and oxygen. And we saw that in the last slide. Remember, he shined, or in a couple slides ago in the experiment, he shined the light onto the algae, and remember, it produced oxygen. And this is why, because in some forms of photosynthesis, oxygen is going to be a byproduct. So photosynthesis doesn't occur in one reaction, though. It occurs in a series of steps in 2 stages. And so the 2 main stages of photosynthesis are light-dependent reactions and light-independent reactions. So the light-dependent reactions are the first stage of photosynthesis. And this is the photo part of the reaction. So photo means light, and so this is light energy. This is where light energy is converted into chemical energy in the form of ATP and NADPH. So the light-dependent reactions occur at the thylakoid membrane. The second part are light-independent reactions. And so this is the second stage of photosynthesis. And this is the synthesis part of photosynthesis. And it occurs in the stroma of the chloroplast. So in light-independent reactions, ATP and NADPH formed in the first part are used to assemble sugars from water and carbon dioxide. And so again, remember this ATP. So the ATP is kind of the universal currency for cells. And so that's what it uses for energy. And so we're going to see this in cellular respiration as the sugars are converted back into ATP so we can use for energy. So ATP isn't really a good long-term storage. And so that's why plants use the short-term ATP to create carbohydrates which they can store for very long. So remember, sugars make starches, so it's long branch chains of sugars, and this is long-term storage of energy. So in light-dependent
in light-independent reactions, that ATP is then converted into the sugar. So again, you create the ATP in the light-dependent reactions, and then you use that ATP to create the sugar in the light-independent reactions. And that's just kind of a general overview of what's going on for those two types of reactions. So in a little more detail, the ADP and NAD plus molecules in the reactions above are low energy molecules, whereas the ATP and NADPH are the high energy molecules. So I told you in previous lectures, the ATP is a currency that cells use for all their metabolic reactions. And so that's why they produce these, and then they use the sugars for long-term energy. So, get into a little more detail in the next couple slides. So the light-dependent reactions in the first stage of photosynthesis convert that energy of light to the energy of chemical bonds. So again, it's the photons going to the bonds that are found in ATP. So there are two different sets of light-dependent reactions. There's a non-cyclic pathway and a cyclic pathway. So again, these are the light-dependent part of the reaction. So light harvesting complexes in the thylakoid membrane absorb photons and pass the energy along the photosystem. So photosystems are clusters of hundreds of chlorophylls. They also contain accessory pigments and other molecules. And these photosystems then release the electrons in the electron transport chain. So think of the light harvesting complexes as arrays of solar panels. So these harvesting complexes collect the sun's rays and then funnel them to a collecting point, which are, so light harvesting complexes are the solar panels which collect the sun's rays, and they funnel them down to the photosystem. So, and this is more of a complete picture of what's actually going on. So electrons are released from the photosystem too. So again, this is the non-cyclic pathway of the light-dependent reaction. And so this is more of the picture that I want you to remember. It's kind of confusing, the first part. This picture, I know it looks confusing, but it actually simplifies what's going on. So electrons are released. You can see the light energy hits the photosystem too. This excites the electron, and we'll get more into this in a second, but it's actually exciting an electron from water, and it's in that process breaking apart water to form oxygen and two hydrogen molecules. So then that that electron is now high energy, and it goes down the electron transport chain, and the energy from that electron is transferred and used to pump hydrogen ions across. And so you're going to have hydrogen ions pumped across into the inside of the thylakoid compartment, and you're going to increase the hydrogen ion concentration. So that increases the amount of hydrogen ions. So then light energy is used to hit photosystem 2. And these electrons, again, so now you have high energy electrons. They're going to bounce down the electron transport chain. And this is going to be used to convert NADP, which is a low energy molecule, combines it with hydrogen. So this is reducing this molecule and storing energy. So NADPH is now a high energy molecule. At the same time, to slow down its concentration gradient of the hydrogen through this channel, which is called an ATP synthase. And that energy is used to combine ADP, which is, again, a low energy molecule, and a phosphate, into ATP, which is a high energy. So this is the non-cyclic pathway, and you'll see why in a second when I compare it to the cyclic pathway, because the electrons are used and you're going to replenish it by breaking apart water. So in order to do the non-cyclic pathway, you need to have an input of water to furnish those electrons. And also something to note, non-cyclic pathway, you're going to have this byproduct of oxygen being produced. So this slide just shows the same thing that I just went over, that photosystem 2 replaces electrons that are lost to the electron transport chain when they're energized by pulling them from water. So again, this dissociates the water into hydrogen and oxygen. And the process of breaking apart the water is called photolysis. Again, if you break apart that word, photolysis, you have light causing lysis or breaking. And so photolysis means you're using light to break apart the water. Okay, and again, this slide, same thing. This is just what we went over, going through the process of electron transfer phosphorylation. That's just talking about using the energy from this high energy electron as it goes down the electron transport chain to phosphorylate this ATP. So this is just a summary of what we went over, so I'll go through it again. So the steps in the non-cyclic pathway of light-dependent reactions. So the first step, light energy ejects electrons from photosystem 2 into the electron transport chain. Photosystem 2 pulls replacement electrons from water molecules and breaks apart oxygen, breaks apart the water molecule into oxygen and hydrogen ions. And so the oxygen leaves the cell as O2. So energy from the electrons in the electron transport chain pumps hydrogen ions, so you can see that here. So it pumps it from the stroma into the thylakoid compartment, and a hydrogen ion gradient forms across the thylakoid membrane. Then light energy ejects electrons from photosystem 1. So what's happening is you have an electron here, the light energy excites it, it bounces down the chain and goes to photosystem 2. So again, low energy here at the bottom, it's excited to high energy, it transfers or uses up that energy to pump hydrogen across, and now it's low energy again when it gets to photosystem 1. So the energized electrons then move through a second electron transport chain when light energy hits photosystem 1. And that energy is used to cause NAD+ to combine with hydrogen ions and form NADPH. So hydrogen ions in the thylakoid compartment then diffuse through the interior of ATP synthase, that's this right here, and across the thylakoid membrane, and it's this hydrogen flow down the gradient through the ATP synthase that causes ADP to combine with a phosphate and form ATP in the stroma. So now you'll see the difference with the cyclic pathway. So in the cyclic pathway, electrons released from photosystem 1 enter an electron transfer chain, but then instead of being lost these electrons cycle back to photosystem 1. So because of this, so in the cyclic pathway, there's only photosystem 1. Whereas in the non-cyclic, remember we had photosystem 2 and photosystem 1. So with the cyclic pathway, you're going to excite the electron, and it's going to cycle back to photosystem 1. And so you don't need to replenish these lost electrons. And so therefore, what byproduct are you not going to have? So if you don't need to replenish electrons, remember, photolysis broke apart water to resupply those electrons and created byproducts. So if you're not going to need to replenish the electrons on the photosystem, what byproduct would you not end up having with the cyclic pathway? Exactly. So you would not have oxygen as a byproduct. So for cyclic, you don't have oxygen. So something else, too, that you don't have for the cyclic pathway is you're not going to make NADPH. So NADPH, remember, is another high-energy molecule besides ATP. So you still are going to form ATP with the cyclic pathway, but between the cyclic pathway and the non-cyclic pathway, which do you think is going to produce more energy? So on the non-cyclic pathway, you're going to have both the high energy molecules ATP and NADPH produced. Whereas with the cyclic pathway, you are only going to have ATP produced. And so therefore the non-cyclic pathway is actually can produce more energy. So energy flow and light-dependent reactions is an example of how organisms use energy harvested from the environment to drive cellular processes. So the simpler cyclic pathway evolved first, and so this operates in nearly all photosynthesizers. So some organisms, though, have became modified to add a photosystem too. And they begin a sequence of reactions that removes the electrons from water molecules and releases the hydrogen ions and the oxygen. So photosystem 1 was the first process, or the cyclic pathway was the original photosynthetic process. And as plants evolved, they evolved this more, this second photosystem that then produces more energy. So having alternate pathways in the same organism is more efficient because cells can produce NADPH and ATP or produce ATP alone. So NADPH accumulates when it's not being used, and this can back up the non-cyclic pathway because NAD plus is not regenerated. So the cyclic pathway predominates in this situation. So if you have a buildup of NADPH and you don't have enough NAD plus available, you're going to do the cyclic pathway because NAD P plus isn't necessary for this pathway to go. So when sugar production is high, NADPH is usually used quickly and converted back into NAD plus. And so therefore it does not accumulate. So therefore in this case, where sugar production is high, the non-cyclic pathway would predominate. And so the organism can switch back and forth between the two processes depending on the condition. And so in more evolved organisms, you're going to have both the cyclic pathway and non-cyclic pathway actually in the same plant. So it looks like, Justin, you have a question? Correct. So the cyclic pathway only has photosystem 1, whereas the non-cyclic pathway has both photosystem 2 and photosystem 1. So here, let's go back a couple slides, and so you can see it here. This one, clear this. So this one on the left, the cyclic pathway, you only have photosystem 1. Whereas on the non-cyclic pathway, you first hit photosystem 2, and then that excites the electron, which then goes to photosystem 1. So the cyclic light-independent reaction of the Calvin-Benson cycle. So this is, remember now this second part is light-independent reaction. So even though I'm using the word cyclic, I'm actually referring to the second part of photosynthesis, not the cyclic versus non-cyclic parts of the light-dependent reaction. So these are light-independent reactions, and it's called a Calvin-Benson cycle. And so this part, the light-independent reactions, remember, is the synthesis part of the photosynthesis. And this is where carbon fixation occurs. And so the carbon is used to produce sugars. So inside the stroma, the enzyme Rubisco attaches the carbon from carbon dioxide to RUBP to start the Calvin-Benson cycle. And so I'm not going to make you learn a lot of this stuff. You don't need to know every part of the photosynthetic pathway, and especially the Calvin-Benson cycle. You need to understand main kind of ideas, and one guide to what you need to know and what you don't need to know are the handouts that I have. So make sure you understand everything that's on the handout. So especially that the light-independent reactions is where carbon is fixed from carbon dioxide. But you don't need to know all the steps of the Calvin-Benson cycle. So the Calvin-Benson cycle is are the light-independent reactions of photosynthesis. And again, this is the cyclic carbon-fixing pathway that forms sugar from carbon dioxide. So carbon fixation is the process by which carbon from an inorganic source, such as carbon dioxide, gets incorporated into an organic molecule like sugar. So Rubisco, or it's another name for ribulose bisphosphate, carboxylase. This is the carbon-fixing enzyme of the Calvin-Benson cycle. So Rubisco is an enzyme that is going to take the carbon from carbon dioxide and start the process of forming a sugar. So we use the term fixation, and we'll come back to it later too in the class. The idea of fixation is taking a gas and converting it into an organic molecule or a chemical. So another area where it's used is nitrogen fixation in plants. And this is where nitrogen gas is pulled out of the air to be used by plants. So it's a conversion from a gas into a chemical that the plants can use. So as was stated in an earlier slide, ATP and NADPH are produced in the first set of reactions, the photo portion or light-dependent reactions. And the light-independent reactions then use the energy from these molecules so the ATP and the NADPH, in order to synthesize sugars from carbon dioxide. And that second part, going from ATP and NADPH to sugars, is the synthesis portion of photosynthesis. So there are four main steps in the light-independent reactions of photosynthesis. So first, 6 carbon dioxide enter the chloroplast, and rubisco attaches each to an RUBP molecule. And so this results in the resulting intermediate splits and 12 PGA are formed. So next, each PGA gets a phosphate group from ATP, and so this is where the energy is that was produced in the previous. So you need this input of ATP. So plus hydrogen and electrons from NADPH and the 12 pGal are formed. So 2 pGal then combine to form 1 glucose molecule. And then finally, the remaining 10 pGal regenerate the 6 RUBP and use energy from ATP to be used in the next round of the Calvin-Benson cycle. And so you can see there's 2 areas on this where you have an input of ATP. So in this step, you have 12 ATP, and in this step, in step 4, you have another 6 ATP that are being used. And so that's the input of energy to be able to produce the glucose and then regenerate these RUBPs to keep going in the cycle. So again, like I said, you don't need to know all of the parts of the cycle. The main thing I want you to understand, you have an input of carbon dioxide, an input of the energy from the ATP, and this produces glucose. And so something else I want you to understand, the word glucose is a molecule, it's an organic molecule that has carbon in it, and that carbon is coming from this carbon dioxide. So you're fixing the carbon from the gas carbon dioxide into a glucose molecule. So when environments differ, so do details of the light-independent reactions, and there are 3 pathways of sugar synthesis. And so what I just showed you is the general, that's the Calvin-Benson cycle. And so there are just a couple variations, and these variations are C3 plants, C4 plants, and CAM plants. And I want you to understand what the difference between these different plants are and why you would use one, or why a plant would be one type versus the other. So there are 3 types of plants because of the process of photorespiration. So on dry days, plants are going to conserve water by closing their stomata. And stomata are the vents that allow gases and water to pass into and out of the waxy leaf. So leaves have a waxy waterproof coating on the outside of the leaf to prevent them from drying out. And so unfortunately though, in order to conserve water, it has this waxy coating, but the waxy coating doesn't allow gases to exchange. So that byproduct of oxygen that you produce can't leave the leaf, and CO2 that you need for the synthesis part, the light-independent reaction, can't enter the leaf. And so you have these little pores that can open and close to kind of play what's more important of either preventing water loss or allowing the exchange of the gases. So again, when stomata are closed, O2 from photosynthesis can't escape, and carbon dioxide for photosynthesis can't enter. So in C3 plants, high O2 levels cause rubisco to attach oxygen to RUBP instead of carbon dioxide. Remember in the Calvin-Benson cycle, we want to attach the carbon dioxide to RUBP. But if there's too high a concentration of oxygen, the rubisco can't tell the difference between the oxygen and the carbon dioxide. And so it's going to attach this oxygen instead. And unfortunately, this pathway is called photorespiration. This is going to reduce the efficiency of sugar production on dry days because glucose can't be produced from oxygen. It needs carbon dioxide to be produced. So if oxygen combines with the RUBP, so if you have oxygen being combined with RUBP, it forms glycolate. And then you're going to need to expend energy to get it back into the PGA form. And so therefore, it's not as efficient of a cycle, and so you're going to have a loss of efficiency of sugar production. Plants get around the process of photorespiration using different processes. So a C3 plant is a type of plant that uses only the Calvin-Benson cycle. So this type of plant isn't as worried about being dehydrated. So there's plenty of water around for a C3 plant. A C4 plant is a type of plant that minimizes photorespiration by fixing carbon twice in two different cell types. And then a CAN plant is a type of C4 plant that conserves water by fixing carbon twice at different times of the day. So both the C4 plant and a CAN plant, they're both C4 plants, but they're just separating the process by two different ways. So a CAN plant does it by fixing carbon at different times of the day. Remember, if the plant doesn't open its stomata until night, it's going to be cooler, and so it's not going to have as much water evaporate. And so that's what a CAN plant does. Whereas a C4 plant, and you'll see in the next slide, this plant just separates the whole process. Actually, I think it might be, yeah, so we'll come back to that. So you'll see in the next slide of how this works. So most plants are C3 plants, and they only use the Calvin-Benson cycle. So like I said, if there's plenty of water around, the easiest solution is going to just be the Calvin-Benson cycle because you're not worried about this photorespiration and less efficient photosynthesis. So whereas C4 plants, the carbon fixation occurs twice. So the first reactions occur in the mesophyll, and the cells capture carbon, attaching it to PEP, and this creates a 4-carbon molecule. The carbon dioxide is concentrated, and then it's released in the bundle sheath near rubisco. So you can see these occur, it's kind of hard to tell in the slide, but these are two separate cells. And so you're separating the process, and so therefore less photorespiration is going to occur. So, and the reason for part of this is because you're concentrating the carbon dioxide in these cells. So unfortunately though, the process of fixing carbon uses energy from ATP, you can see here. And so this is also going to be a little less efficient than just a C3 plant. But at least you're not having the total inefficiency of photorespiration. So one example of a C4 plant is corn. So there's a trade-off of why plants use this kind of double fixation. And so plants that are in an area where they might dry out, this is one way to get around the photorespiration. But again, like I said, it's not quite as efficient as the C3 pathway. So CAN plants minimize photorespiration by opening stomata and fixing carbon only at night. So this limits the amount of evaporation that occurs when the stomata are open. And so one example of this is the jade plant. So the trade-off with this is that plants are only able to make sugar at night. So C3 plants can make sugar any time of the day. So they're a factory that's always open. Whereas CAN plants can only make it can only do this, it can make the sugar at night. So they're only open, say, 8 hours a day. And so the differences between C3, C4, and CAN plants show the alternate pathways that plants have adapted to survive and thrive in different environmental conditions. This allows plants to have a high level of sugar production, whether they're in very wet environments or whether they're in very dry environments. And so this is just one of the many ways that plants have adapted to their environment. So that's the end of chapter 6. So let's go ahead and take a 10-minute break. So we'll come back at 8:10, and we'll go over chapter 7. Actually, before we take a break, are there any questions on what we just went over? Sure. What's the question from the handout? So this question, I just referred to it for a second. So the question is what do the plants do with the glucose to store it after photosynthesis? And this comes back, I think, from chapter 3 when we went over different organic molecules. So plants store energy in starch. So remember, starch is just it's branch chains of glucose molecules. Does anybody remember how humans how humans store glucose? What form do we store it in? Correct, glycogen. Any other questions? Okay, I'll see you in about 10 minutes. Okay, so let's go ahead and get started again on chapter 7. And so like I said, chapter 7, we're looking at cellular respiration and how that happens. So this is how both plants and consumers use the energy that the plants stored in the form of the starches or carbohydrates. So mitochondria produce ATP by aerobic respiration. So electron transfer chains in mitochondrial membranes set up hydrogen gradients, kind of pretty much very similar to the same thing that we saw in photosynthesis with a hydrogen gradient. And these hydrogen gradients power ATP formation. So many disorders related to defective mitochondria are known, including Friedreich's ataxia. And this is a disease where the enzyme building protein frataxin does not work properly. So therefore, enzymes in the mitochondria are not fully functioning, and this causes loss of coordination, ataxia, weak muscles, and heart problems. So this picture shows an electron micrograph of the inside of a mitochondrion. So as you can see, mitochondria have an internal folded membrane. So again, this is a similar idea to what we saw in the chloroplast. So this folded membrane allows for the generation of ATP. So most organisms convert the chemical energy of carbohydrates that they built or that were ingested to the chemical energy of ATP. So anaerobic, this means without oxygen present, anaerobic when oxygen is present. These are both pathways of carbohydrate breakdown. and both start in the cytoplasm with glycolysis. So this is something you definitely need to know for the test, glycolysis, and we'll come back to it many times to just make note of it. Glycolysis happens in the cytoplasm. So glycolysis is the process of converting glucose and other sugars into a molecule called pyruvate. And I do want you to remember this part, that glycolysis converts glucose into pyruvate. So this process of glycolysis does not require oxygen. And so if there is no oxygen present, such as in anaerobic bacteria, the pathway ends with fermentation and yields a total of 2 ATP per molecule of glucose. So these three terms you need to know well. So again, glycolysis is the set of reactions in the cytoplasm in which glucose or another sugar is broken down to 2-pyruvate for a net yield of 2 ATP. So the second term is pyruvate, and this is just a 3-carbon end product of glycolysis. And then finally, fermentation. We'll come back to it later on in the chapter. This is an anaerobic pathway, meaning it doesn't require oxygen, by which cells harvest energy from carbohydrates to produce ATP. So a net of 2 ATP molecule glucose, a net of 2 ATP per glucose molecule are produced. So fermentation is the process that happens when only glycolysis happens. So fermentation is glycolysis in the presence of no oxygen. So the first cells on Earth did not use sunlight for energy. So ancient organisms extracted energy and carbon from simple molecules such as methane and hydrogen sulfide. So these were both gases that were very plentiful in Earth's early atmosphere. So then aerobic respiration, which uses oxygen and yields much more ATP. Sorry, I skipped the slide. So aerobic respiration uses oxygen and yields much more ATP than fermentation. So this process evolved after oxygen was released by early photoautotrophs during the process of photosynthesis. So remember, in those organisms where you had non-cyclic photosynthesis, this produces as a byproduct oxygen. So over time, the photosynthetic organisms released the O2 as a byproduct, and this allowed for aerobic respiration to occur. So O2 became, or oxygen became a major component of the Earth's atmosphere over time. So the general equation for aerobic respiration is glucose and oxygen are converted into carbon dioxide and water. So this is just basically the reverse of the general equation for photosynthesis. So remember, photosynthesis, we took water and carbon dioxide plus light and made glucose and oxygen. So this flow chart shows the connection between photosynthesis and aerobic respiration. So again, the byproducts of aerobic respiration, carbon dioxide and water, so you can see that here, these are the reactants of photosynthesis. And the products of photosynthesis are the reactants of aerobic respiration. So this is a cycling of materials. On the other hand, there's only a one-way flow of energy. So you can see it goes in with photosynthesis, and it goes out of the system with aerobic respiration. So both fermentation and aerobic respiration begin in the cytoplasm with glycolysis. And so just to note, this rectangle is the mitochondria. So this right here is the mitochondria. So anything outside of it is the cytoplasm of the cell. So it's kind of hard to imagine in the slide the way it's set up, but just picture the cell is around like that. So in this area right here is the cytoplasm. So you're still inside the cell. This rectangle is just what they're showing as the mitochondria. So again, both fermentation and aerobic respiration begin in the cytoplasm with glycolysis. So then after glycolysis, pathways of fermentation and aerobic respiration diverge. So in aerobic respiration, this process continues inside the mitochondria. And this ends with oxygen accepting electrons at the end of the electron transfer chain. And the other process of fermentation, this ends in the cytoplasm. So this is where a molecule other than oxygen accepts an electron. That's what fermentation is. So A in the figure is the first stage, glycolysis, and this occurs in the cell cytoplasm again. And so enzymes convert a glucose molecule to 2-pyruvate for a net yield of 2 ATP. You can see that here. So just to note, so remember, we learned about it, I think, in chapter 2, of where chemical reaction needs a little bit of an input of energy to get over the activation energy to then proceed. And so the same is true for glycolysis. You need an input of 2 ATP just to get the glycolysis reaction to start going. So glycolysis produces 4 ATP per glucose molecule, but you have to input 2, and so therefore there's a net of the 2 ATP. So it produces 4, but it's going to be a net of 2. So also in this process, 2 NAD+, which is a low energy molecule, combine with electrons and hydrogen ions during the reaction to form 2 NADH, which are high energy molecules. So D is the second stage, and this occurs inside the mitochondria. And in this stage, 2-pyruvate are converted to a molecule that enters the Krebs cycle. So carbon dioxide forms and leaves the cell, and 2 ATP, 8 NADH, and another high energy molecule, FADH2, form during these reactions. And then finally, T is the third and final stage, and this is the electron transport chain or electron transfer phosphorylation, and this also occurs inside the mitochondria. So in this third stage, the 10 NADH and 2 FADH2 that were produced during the other reactions give up the electrons to the electron transfer chain, and this causes hydrogen ions to pump across the membrane. And so again, similar to what we saw with photosynthesis, this forms a gradient. So then this gradient is used again to drive ATP formation, same as what we saw with photosynthesis. And so in this reaction though, oxygen accepts the electrons at the end of the chain, and unlike photosynthesis where you broke apart water to steal the electron, in this situation you're donating electrons to oxygen and reforming water. So a byproduct of cellular respiration is to actually form water and you use up oxygen. Whereas a byproduct of photosynthesis, you're going to form the byproduct of oxygen by using up water. So again, you can see that in very many ways, cellular respiration is just the exact reverse of photosynthesis. So the general process of glycolysis is shown in the slide. So enzymes use 2 ATP to convert 1 molecule of glucose or another 6-carbon sugar to 2 molecules of pyruvate. So again, in the process, 4 ATP are formed and also NADH is formed. And so this kind of basic thing of what's going on during glycolysis is really the gist of what I want you to know for the test. But just to kind of show you the other steps in more detail. So the first step, an enzyme transfers a phosphate group from ATP to glucose, and this forms glucose 6-phosphate. Next, the phosphate group from the second ATP is transferred to glucose 6-phosphate, and the resulting unstable molecule splits into 2 3-carbon molecules of pGal. Finally, 2 ATP have been invested in the reactions at this point. Then next, enzymes attach the phosphate to the 2 pGal and transfer 2 electrons and 1 hydrogen ion from each pGal to NAD+. So 2 PGA and 2 NADH form, and enzymes transfer a phosphate group from each PGA to ADP. So 2 ATP are formed by substrate level phosphorylation. So the term substrate level phosphorylation just means not from the electron transport chain. So again, you're investing 2 ATP to form 4 ATP in this situation, and so therefore you have a net of 2 ATP and 2 NADH from glycolysis. And so finally, the last part of glycolysis, and so enzymes attach a phosphate to the 2 pGal and transfer 2 electrons and 1 hydrogen from each of the pGal to NAD+. Finally, in this last stage, the enzymes transfer a phosphate group from each of the 2 intermediates to ADP. So 2 molecules of pyruvate form at this last step. So 2 more ATP are formed by a phosphate. The 2 more ATP are formed by substrate level phosphorylation, and so therefore this is where you get your total of 4 ATP that are formed. So depending on the type of cell and environmental conditions, so like I said, whether or not oxygen is present, the pyruvate either enters a second stage of aerobic respiration or it's used in other ways such as in fermentation. So we're going to skip to aerobic respiration inside the mitochondria, and then we're going to come back to how fermentation works later on. So in the second stage of aerobic respiration, acetyl-CoA formation and the Krebs cycle takes place in the mitochondria. Again, I don't want you to know all these specific steps in the Krebs cycle. I just want you to understand kind of the gist of it. So in the Krebs cycle, 2 pyruvate are used to form carbon dioxide and ATP and more high energy molecules, so NADH and FADH2. And something else you don't need to know, you don't need to remember how many of these high energy molecules are formed at each step. So there are 8 steps in the second stage of cellular respiration. So I'm not going to go through all of them. If you're interested, take a look at a lot of biochemistry, and I'm just not making you remember all the different steps of the Krebs cycle. So, but again, a byproduct of the Krebs cycle is carbon dioxide. We'll skip over this. These are all the steps of the Krebs cycle. So the third stage of aerobic respiration is electron transfer phosphorylation. and this occurs at the inner mitochondrial membrane. So this third stage is where most of the ATP is formed. Actually, we'll come back to it, the sum up of how much ATP are produced during each process, but the most ATP are by far are produced in the third step of respiration. So this third step of aerobic respiration is electron transfer phosphorylation. And in this step, coenzymes, this is the NADH and the FADH2, that were produced in the first stages, deliver those electrons in hydrogen ions to the electron transfer chains in the inner mitochondrial membrane. So these electrons move through the chains and release energy. And again, the energy is used to force hydrogen against its concentration gradient so that you have a very high concentration of hydrogen in this internal membrane area. So those electrons moving through the chains release the energy, and that's what's used to move the hydrogen into the inner membrane space. So hydrogen ions accumulate in this inner membrane space and form a gradient across the inner membrane. So again, you're going to have high hydrogen here, low hydrogen inside. And so that's the gradient between the two. So these hydrogen ions then follow the gradient back to the matrix through transport proteins, which are ATP synthases. And so again, this is almost identical to photosynthesis. You're going to have the hydrogen flow back down its gradient through that ATP synthase, and that causes ADP to combine with phosphate and form more of a high energy ATP molecule. And oxygen combines with the electrons at the end. So we have oxygen here combining with the electron to form H2O and more water. So this slide sums up what's going on in each of these steps. To really make sure you understand what's going on in this slide. Like I said, it's basically a summary. But overall, aerobic respiration typically yields 36 ATP for each glucose molecule. So remember, aerobic respiration is only going to happen in the presence of oxygen. So with aerobic respiration, you have 32 ATP forming in that third stage. So the total 36 is kind of an average. This differs a little bit by cell type. So for example, the typical yield of aerobic respiration in brain and skeletal muscle cells is going to be 38 instead of 32. Instead of 36. So you had a net of 2 ATP that happened from glycolysis, 2 ATP that occurs in the Krebs cycle, and 32 ATP from the electron transport chain. So again, what are you going to do though, for one, if there's not enough oxygen present? So for instance, if you're an anaerobic bacterium that grows in an oxygen, in an environment that doesn't have oxygen. So the way that cells get around this is called anaerobic fermentation. And so the anaerobic fermentation pathway begins with glycolysis, and it finishes in the cytoplasm. So this doesn't involve the mitochondria at all. And there are two types of fermentation. and they include alcoholic fermentation and lactate fermentation. So the process of fermentation has a molecule other than oxygen accepting electrons at the end of these reactions. So alcoholic fermentation is the anaerobic carbohydrate breakdown pathway that produces ATP and ethyl alcohol. So this process is used to make wine and breads and pretty much any other alcohol. So lactate fermentation, on the other hand, is an anaerobic carbohydrate breakdown pathway that produces ATP and lactate. So this is the process used in cheeses and pickles. So this is also the process used in your muscles when you exercise too hard and you're not taking in enough oxygen. And this lactic, the lactate that's produced is lactic acid, and the acid is what makes your muscles sore afterwards. So the final step of fermentation, regenerate the NAD+. So remember, in the process of glycolysis, we converted NAD+, which is a low energy molecule, into NADH. So that's great if you wanted to use the NADH molecule in the electron transport chain. But the problem is there's no oxygen present, and so therefore, in order to continue glycolysis, we need more of this NAD+ molecule. So if we didn't do alcoholic fermentation, we would just keep on building up NADH and run out of NAD+. So we need to find some way to regenerate this NAD+ from the excess NADH. And that process is alcoholic fermentation, or actually any type of fermentation. And so, again, the point of fermentation is to regenerate NAD+, so at the very least we can continue on with glycolysis. So even though glycolysis only produces 2 ATP, 2 ATP is better than nothing. And especially if you're a small organism, 2 ATP might be enough to suit your needs. So in a human, you're going to need a lot more energy than you would if you're a small organism. And so therefore, that's why we rely on cellular or oxidative phosphorylation, and so we had to have oxygen to survive. Whereas anaerobic bacteria can survive by just producing NAD+. Can just survive on producing 2 ATP. So we can use this process short term, but in the end we need to go back to producing large amounts of ATP. So skeletal muscle has 2 types of fibers, red and white. So ATP is produced primarily by aerobic respiration in red muscle fibers, which sustain activities that require endurance. So the red appearance, which causes the dark meat that you eat on Thanksgiving, is from myoglobin. So this is a protein that's similar to hemoglobin in your red blood cells, but instead myoglobin concentrates oxygen in the red muscle fibers for aerobic respiration. So lactate fermentation in white fibers supports activities that occur in short, intense bursts. So things like if you're going to go sprinting versus a marathon. So this is the white meat, such as the turkey breast meat. And people are born with different proportions of white and red muscle fibers. And so this proportion can make people more suitable to sprinting versus long distance running. So a lot of times, it doesn't matter how much you train to run a marathon, you're better off or you're more suited to run sprints and vice versa. So now we're going to switch over to a little bit of a different look at it of how does your body use other foods besides, so we talked about just using glucose, but how does your body use the protein that you eat and the fat in the starches in order to convert into ATP. So remember, we can't use any of those other foods that we take in in our metabolic reactions. Our body runs off of ATP. So we need to convert all that stuff into the usable energy of ATP. So and your body's able to use the energy stored in the chemical bonds of most of the foods that you eat. So simple sugars from carbohydrate breakdown, glycerol and fatty acids from fat breakdown, and carbon backbones of amino acids from protein breakdown can all enter the aerobic respiration pathway at various reaction steps. So when energy demand is low, ATP concentration rises, and liver and muscle cells store glycogen. So they store excess glucose. So whereas when you start using it, so if you're exercising, glucose levels are going to decline, and the pancreas secretes glucagon, which causes the liver cells to convert glycogen back into glucose. So this is the first energy reserve the body taps. So glycogen makes up about 1% of an adult's total energy reserve. So again, glycogen is a fast way to regenerate your glucose reserves to be able to feed in the glycolysis. So your body gets energy from fats that you ingest or that you have also stored. So if you don't have enough glycogen to support your energy needs, your body can also use some of the other stuff that you eat and also the fat that you've stored. So when blood glucose gets too high, excess dietary carbohydrates are converted to fatty acids, and the body stores most fat as triglycerides. So triglycerides have 3 fatty acid tails, and then the reverse happens when blood glucose falls. So triglycerides provide energy, the glycerol is converted to an intermediate of glycolysis, and the fatty acids are converted to acetyl-CoA, which if you look at the Krebs cycle is what goes into the Krebs cycle. So when you eat more protein than your body needs, the amino acids are broken down, and the amino group is going to be removed and become ammonia. So the carbon backbone is then split, and acetyl-CoA, pyruvate, or an intermediate of the Krebs cycle forms. And so these last couple slides were just showing that all these different things that you eat can enter cellular respiration at different points in that pathway. So again, the triglycerides from fats can enter into the Krebs cycle, and then proteins can be broken down and can enter different parts of the Krebs cycle and cellular respiration. Anybody have any questions on Chapter 7? So, as I stated before, it's a lot of information in Chapter 6 and 7. Really focus on the handout, because that's what I want you to know. What's on those handouts. And if you do have any questions on the handouts, I'm more than happy to answer questions. If you can't find, so most of what I ask should be in the lectures. If you can't find it there, it should definitely be in the book. And if you're still having problems finding it, feel free to just send me an email, and I'll help you out with it. One thing I do not want, though, is for you to just basically email me questions to have me do your handout for you. But other than that, I'm more than happy to answer questions.